zigzag pattern so that the cells would form capillary network in this pattern, and as the pattern grow, as the capillaries grow, we can quantify how much angiogenesis has happened in the bioprinted tissue. So the way this works is we start with a scaffold made of PLG. I'll come back to the scaffold in the third part of my talk. We start with a scaffold. We flip it, and we print on one side with the bio ink, like I just showed you, mixed with hydrogel mixed with cells. We flip it upside down, and we see the RB cells on top, and then we let it mature for 4 to 5 weeks, and the scaffold degrades, and RP and endothelial cells replace that space with an ECM that we call kind of a Brooks membrane equivalent. So it becomes really a three-dimensional tissue that should really look like the back of the eye. And in fact, if you look at the tissue, that's how it looks. And really, RP cells on top, and an area in between, which we call Brooks membrane, where the cells have secreted ECM, and it really contains most of the component of ECM component of Brooks membrane, and a capillary network in the back. And you can see the capillary network here marked in green and red is made more closer towards the scaffold, towards the RPE, and it's more dense towards the RPE, just like how it is in the choroid, and less dense further away from it. So if we zoom through the entire tissue, you can see there are capillaries of various sizes, and some actually span from the entire tissue. And this is stained in green for endothelial cells, and in red for pericytes. And you can see nice capillary network from top to bottom. This one is going all the way through tissue, and they have a lumen, which means it's about 20 to 40 micrometer in diameter. And RP cells nicely polarized on top of this 3D tissue. Here they are stained in green with azurin, which is the marker for apical processes, and also an indicator of RP polarization. And in red is marked for E-cadherin, which marks this RP tight junction. So it shows you RP cells nicely polarized on one side, and on the other side there's a nice capillary network, as you see with a red stain marked for PLVAP, which is a marker for capillary fenestration. So fenestration is a very interesting and important feature of choroidal capillaries. What it is that it's known that there's a kind of a small pore in the membrane of endothelial cells in the choroid. These pores are about 70 to 100 nanometer in diameter, and these allow for easy flow of macromolecules between the blood through the Brooks membrane into the RPE, and that's why these choroidal capillaries are called fenestrated. So while we do see high expression of fenestration marker in our printed capillaries, we wanted to make sure that these capillaries are indeed fenestrated. So we looked at them by TEM. It's an example of a published capillary from human fetal RP, so from a cadaver eye, TEM, and you can see this is what the fenestration is. These arrows mark these pores in the membrane of endothelial cells. Now compare that to the pores that we see in our capillaries made with iPSC-derived RP and iPS-derived endothelial cells, and I would say they're pretty similar. We get a very similar pattern of fenestration in the capillaries that we make in our 3D tissue. So we're very excited that we can also make tissue that is fenestrated and behaves similar to the choroid of the eye. And also that we think that fenestration in this tissue is dependent on the presence of RPE cells. Because in this slide, what you see is we made this tissue in the presence of RPE or in the absence of RPE. And in the left, you see a 3D rendering of a small piece of tissue, which is about 100 by 100 micrometer in size. You see RPE cells in the background. And on top, you see high expression of fenestration marker and big and thick vessels. Whereas on the left side, on the right side, you see tissue without the RPE. And you can see the vessels are much thinner and they don't proliferate as much. And the expression of PLVAP is much weaker in this cell, so the expression of the fenestration marker is much weaker. And this was a very small piece, but if you look across the entire tissue, across the entire well, which is about 6 by 6 millimeter in size, you can easily see much weaker proliferation with the absence of RPE and much weaker expression of the fenestration marker. So we think both the capillary growth and the expression of fenestration is dependent on RPE cells. So this is exactly what happens in dry AMD. In the absence of RPE cells, capillaries die off, and it's also thought that at an earlier stage of AMD, the capillaries lose fenestration as RPE cells are not functioning properly. So we think we have created a model for dry AMD in a 3D model for dry AMD that has both RPE and choroid. So the next challenge for us was to create a model for wet AMD. And here's an example again of a wild-type RPE and wild-type endothelial cells, 3D rendering of the tissue from top to bottom. You see RPE, and then you see endothelial cells, vessels labeled in red, going up again, vessels, and then RPE cells. And then what we're going to do is we're going to do what we call a depth scan. So we're going to add pseudocolors to the tissue. And depth scan is you see anything that is at a choroid level is under the choroid is blue, choroid level is green, as you see most of the vessels are. Oops. As you see, most of the vessels are green at the choroid level. And if they're at the Bruch's membrane level, they're yellow. And if they're at the RPE level, they are red. So you can see most of the vessels are green. Some are yellow, and there's no vessel that is red. Now, if I go to the next tissue, which we overexpress a transcription factor called STAT3 that we know causes inflammation in cells or increases expression of inflammatory cytokines, pro-angiogenic cytokines, including VEGF and other interleukins and interleukin 6 especially. What we see is that if you run this 3D rendering, you see RP and then vessels, and then going back up, vessels and RPE. And now if you run the depth scan on this tissue, what you will see is these vessels that are green, but you'll see a lot more vessels that are yellow, so that are up here. And if you focus on this white circle, there's one vessel that goes all the way from green, yellow, and then it turns red. So that means there's one vessel that has gone completely in the Z direction from the choroid into the RPE, just like what happens in case of wet AMD. There's more vessel growth, proliferation in the sub-RPE zone, and there are some vessels that go through the RPE and leak fluid in the retina. And we see exactly something similar happening in our 3D tissue in a dish. So we think we've also created a 3D wet AMD model. So we're using both of these models to really further understand how disease progresses and what drugs we can use to kind of undo some of the hallmarks of AMD, at least in a dish. So to summarize this part of my talk, we have replicated early hallmarks of dry and wet AMD in a 3D disease model using patient-specific iPS, RPE, and endothelial cells. We have structurally and functionally validated these models. I didn't have much time to show you all of that data. And now we're using these to discover some of the genetic pathways that lead to different stages of AMD in patients. Now, in the next few minutes, in the third part of my talk, I want to show you our approach to developing a cell therapy for AMD using patient-specific iPS cells. So the focus in this case is going to be on dry AMD. And what happens in this case is that because RPE cells die, photoreceptors start to die, and as a result, patients go blind. But if you look carefully in patients or in cadaver eyes, what you notice is between the central part of the eye where patients are blind, where photoreceptors are dead, if you go towards the healthy area, there's this area called the transition zone, where photoreceptors are not quite dead yet, but RPE, as you can see, is gone. But these photoreceptors are destined to die. They're probably not functioning properly, and over the next several months of patient's life, these photoreceptors are going to die off. And the disease is going to keep progressing further. So the idea surgeons have that if you can bring with a patch of RPE in the transition zone, you could protect the overlying photoreceptors from dying and stop the disease from progressing further. In fact, this has been surgically tried where people have moved a piece of RPE from the periphery of the same patient's eye into the center, and have seen that in small number of cases where the RPE cells, where the RPE patch integrates into the back of the eye, patients' photoreceptors are repaired and the vision is stops from getting worse. And that is a very important proof of principle for us to really develop an autologous cell therapy that if we can make a patch of RP from patients' iPS cells, patients on stem cells, then we could really stop this disease from progressing further and getting it worse. And that's what we're trying to do. There's a 4 by 2 millimeter RP patch on a PLGA scaffold, and we choose cells on top and the scaffold at the back. And I'll show you all the preclinical studies that we've done, how the scaffold works, to make the scaffold and to test the scaffold in animal models. So an autologous IND requires that a clinical grade RP patch is made at least from 3 patients, and it's tested both first using in vitro GLP studies, functionality of the patch to compare how the cells perform starting from different donors, and then to show with the GLP compliant fashion with preclinical animal studies, safety studies, to see that the cells and the patch is safe and doesn't cause any toxicity, and then to show efficacy in one of the animal models, either a rodent or a pig. So these are the studies that we have performed for our in preparation for our IND application, where we start with the clinical grade. We first optimize manufacturing of clinical grade iPSRP patch and made clinical grade RP patch from 3 clones, 3 iPSC clones per patient, developed the protocols and SOPs to make this patch. And then we tested the functionality of this patch from all 3 donors and have tested now safety and toxicity at 4 different time points, cells from 2 different patients in almost 450 different rats. This study is completed and have tested efficacy of this patch in a rodent model, and are now currently testing efficacy of this patch in a pig model. So all of this data together leads to an IND application that we are putting together. So I'll show you, in the interest of time, I won't be able to go into details of all of this, but I'll show you small highlights of each of these studies. So first, a protocol to manufacture a clinical grade patch. The process is long. It takes about 6 weeks, starting with draw blood from patients, day 0 all the way to manufacturing of patch. We go through process of isolate blood, reprogramming the blood cells into a small bank, into iPS cells that we convert into a small bank of iPS cells at passage 10, which gets validated, and differentiation starts after passage 11. And we go on to making the cells, RP cells, similar, using the process I showed before as well, going through RP progenitor stage, seeding the immature cells at the scaffold, and making mature RP patch on the scaffold. Because the process is so long, we had to have, process go-no-go points, which we have put in place, starting with a bank of iPS cells which get characterized, and then during the differentiation process, we check that the cells are differentiating well, or if they're pure, and then finally confirming the sterility, identity, and purity of cells on the patch. And if the process fails, we have cryopreservation steps throughout the process that allow us to go back to one of these stages and manufacture the patch from this intermediate stage. So this is the scaffold that I showed you earlier too. It's a 400 nanometer diameter electrospun nanofibers of PLGA, and we make it such that our B cells can form a monolayer on top of the patch, which I show you here in the next picture. This is a scanning EM of our B cells grown on a patch. In blue, you see the patch. In brown, you see our B cells and a nuclei of the cells from the side. Because you're looking at it from the side view, you see these apical processes of all the cells in green, and you can see all the cells in the patch grow homogeneously, they differentiate homogeneously, and have similar apical processes, and they're fully polarized. So then we went on to functionally characterizing this patch made from 3 different patients, 3 different AMD donors, and almost 3 clones per donor, performed using GMP-grade protocols. Again, in the interest of time, I won't go into the details, but we checked at differences between patches from different donors, different clones at molecular level, morphological level, and functional level. and using all of these assays that I've listed on the site. This manuscript has been submitted, so it will be out soon. But the goal was here to, first of all, functionally validate the patch. Second, to see how much is the variation, technical variation from clone to clone within one patient. And third is to see how much is the variation from patient to patient. And finally, to identify some of the release criteria from some of these six assays that we're performing to come up with a final way to functionally validate the patch before transplantation as we manufacture it in GMP suites for patients. So all of this work is begun. We have picked some of the molecules, especially transepithelial resistance and geometrics, as potential candidates for release criteria. And I want to show you some of the data we have in animals to test this. We have tested safety and efficacy of this patch. So first thing in animals, we're going to test whether the patch is safe. One of the issues people have, or concern people have, is if any stem cells are left in the final product, that could lead to tumor, teratoma, or another tissue formation in the wrong place, and you want to avoid that. So we, of course, demonstrate that iPS cells, there are no iPS cells left in our final product, and these iPS cells, and that iPS cells do not survive in our final product. But then to test it in animals, whether RP cells would actually, iPS-derived RP cells would actually lead to tumor or teratoma formation, we used immunosuppressed, immunocompromised rats. And this is an example in which we injected pure iPS cells in the subretinal space. And as expected, within a few weeks, these cells form massive teratoma, as you can see in the slide here, with cells of various lineages in this teratoma that can be identified. But if we transplant an RPE patch made from iPS cells, we don't see, and this is, as I said, the study is done over 450 different rats. Not a single rat did we see any tumor, teratoma, or any other kind of tissue in the eye or anywhere else in the body. But interestingly, as the scaffold degrades within a few weeks, the whole patch, as you can see here in the marker, nicely integrates into the back of the rat eye. So this tells you that if the patch transplantation is done, it will allow the entire model layer to integrate and start functioning right away in the back of the eye. So, but to really test how things would look in an AMD patient, and to test the big piece, the 4x2 millimeter patch, we used pig as a model. The advantage with pigs is pig eye is very similar to human eye and in size and anatomy, so we could really test the tool, the surgery procedure we want to optimize for patients for transplantation of this patch. So, but the problem with pigs is that there's no genetic model that's similar to what happens in AMD patients. So to create that, we use laser ablation. So we use a micropulse laser ablation of RPE. We use a micropulse laser, the wavelength that is absorbed only by RPE cells, and as a result, these cells heat up and they die off, and the photoreceptors will die, which you can see here. The bottom panel by infrared image of the eye is a laser damage RPE. You can see because RPE is gone, now the infrared can shine through the eye, so you can see the back of the eye. And in the right side, you see histology H&E stain, where you can see RPE cells are gone, some cells are clumped up, and as a result, photoreceptors are slowly dying. So the idea would be to transplant a human RPE patch here and protect the overlying photoreceptors from dying. So we developed a tool that can bring the patch to the back of the eye. The tool has a curvature that fits the back of the eye. It takes a transplant only in one orientation, and then it can be controlled by a foot pedal, so the surgeon can maneuver easily the patch in and out and deliver it to the right place. So it's a surgery that shows you how the tool is delivered to the back of the eye. The pig eye has been prepped, and the surgeon is making the incision bigger to bring the tool inside. And in the next slide, the surgeon is going to deliver the tool, bring the tool here. The tool contains the patch. There is a cut in the retina. Retina has been lifted right here. Surgeon is aligning the tool tip to the cut of the retina and slowly will deliver the patch under the retina. And then the retina is flattened on top of the patch. and the patch stays right under the retina, which we can prove by non-invasive imaging of the eye by a technique called OCT, optical coherence tomography. You can see from the top view is a patch, and from the side view is a patch under the retina shown here by yellow line, and retina looks fairly healthy on top of the patch. So now to show that the patch is really functioning and is providing efficacy, we use a technique called multifocal ERG. So basically we shine multiple light pulses to the back of the eye, and depending on where the light pulse hits, it gives us an electrical response from that part of the eye. So it tells us about the health of that part of the eye. For instance, if the light pulse hits the healthy part of the eye, it gives us this nice and robust big electrical peak, so it tells us that part of the eye is healthy. But if the light hits this laser injured part, which is right here next to the patch, and the patch is not there, and you can see there's almost no response, suggesting that because of laser injury, photoreceptors are not dead, and retina is dying. But if the light hits the transplanted part of the retina, where we put the human patch, you get a response that is in between, suggesting that the human RB cells are able to protect the pig retina, pig photoreceptors to some extent, so that we can get some electrical response from that part of the eye. By histology, we can show that the human patch integrates even in the pig eye, just like the red eye. You can see here human cells are labeled in green, and both pig and human retina, both pig and human RP are labeled in red here. You can see pig RP is labeled with P65 RP maturity marker, nice and red label, and as it enters, this part enters to the human patch, which is also green, you can see these cells are starting to mature, and they're pretty much in direct continuation with the pig RP. So they nicely integrate into the back of the eye, and they also protect photoreceptors, as can be confirmed by histology. If we compare these two stainings, this is from the same eye, area that was lasered but no transplant was given, versus area that was lasered but the transplant was given, which you can see right here. Compare the thickness of a photoreceptor layer, photoreceptor nuclear layer, in stained here by dark blue. It's almost double the thickness of the area where there was no transplant, suggesting that human cells are able to protect photoreceptors from dying in the pig eye. So with that, I want to conclude and show you that we are towards the end of this project. We've been doing IND enabling studies for the past about 4 years. In green is everything that we have almost finished, which I showed you data on, tissue sourcing to generating GMP-grade iPS cells, and performing validation of these cells and preclinical safety and efficacy studies. We're now in the process of writing our IND, and we hope to submit that soon. And I want to thank the entire team. And with that, I'll be happy to take any questions.
Great. Thank you so much, Dr. Bharti. This is a really great presentation, so thank you so much for taking the time today. So for those listening on the GoToWebinar, you can take the next couple minutes here to submit any questions that you have for Dr. Bharti. There's that little question box where you can submit those. While those questions are coming in, do you want to switch over to the other presentation, Marissa? Okay. So while those questions are coming in, I would like to mention to everyone that CDI is now offering human iPS-derived retinal pigment epithelial cells. So these cells are provided as a cryopreserved vial of cells. We have 2 different unit sizes, a smaller 1 million vial and a 5 million vial, as well as a pre-packed kit. So these cells are a very pure population of cells, at least 80% positive for BEST1, and they are, reproducibly produced, ensuring the same performance with every batch. So I just did want to mention that. We also have a program right now where we are awarding 3 vials of human iPS-derived neural cells. There's 6 different cell types actually that are part of that grant program, including the ISL retinal pigment epithelial cells. So this is out there to support innovative research, and if you'd like to enter, you can navigate to our website at cellulardynamics.com/grants. just provide a brief description of your research aims and how CDI's cells would be used in your work, and winning applications will be awarded a free vial of cells to get your project started. Okay, so let's check in here and see what questions we have that have come in. Just 1 minute here while I pull that up. If we aren't able to get to your question in the next, looks like we have about a little over 10 minutes here left of the webinar, we will reach out to you, our support team will reach out to you directly with the email that you've provided so that we make sure that we get your questions answered. But hopefully we'll see if we can get to as much as we can here. Dr. Barty, so have you examined any use of your approach for RPE damage associated with retinal detachment in addition to AMD? So retinal detachment causes general and extensive damage to the RPE.
No, we haven't. The simple answer is we haven't tested it in any other condition. In fact, we have only tested it in a pig animal model that we showed for laser injury-induced RPE damage. And we tested it in a rat animal model in which RPE is dysfunctional due to genetic mutation, and that leads to photoreceptor cell death. So there are only 2 cases where we have tested the efficacy of these cells, and they work in both cases. I don't see a reason why it would not work in that case. If it's done it well in time, before when the retina is still healthy, it should work and should help the attachment of the retina in case of retinal detachment, but we haven't tested it yet.
Great, thank you. Another question here, more towards looking at the actual differentiation of RPEs from iPS cells. So we have someone interested in the protocol of being able to grow RPE cells from iPS cells in as little as 10 weeks. Previous methods took more like 6 months to create RPE clones from iPS cells. Has the process been accelerated, or are there different types of RPE-like cells that are made from iPS?
I would say the process has been accelerated. That's what I showed in one of the first few slides, where we show a schematic of how RPE cells are made. Because in the past it took 6 months because people use a process called spontaneous differentiation, so let stem cells just randomly differentiate into any cell type, and including they would make RPE. But the process that we have optimized uses what is called directed differentiation, where we add specific growth factors at specific time points, guided by really all the animal studies that have been done to study development of the RPE. So really that causes cells to become RPE progenitors at a much earlier time point, and then eventually become RPE much faster.
Great. Okay. Another question about the scaffolds that are used for the transplanted RPE patches. So, do the RPE scaffolds form scar tissue, and when you transplant the RPE patches, do they form scar tissue?
So the one example I showed you of the rat, immunocompromised rats, in which we are able to keep the cells alive for a long time. As you can see, there's absolutely no scar when the scaffolds degrades, and in fact the whole monolayer nicely integrates. So we think if there's no immune response against the cells, there should be no scar tissue formed, and that's the advantage of using autologous cells, that there will be no immune response against the cells.
Any other? Simon, are you jotting down a question over there? No, you're good. I'm just waiting a little bit longer here to see if we have any other questions coming in.
Sure.
Okay, so what is your opinion regarding using cells in suspension versus cells on a scaffold?
So I think it depends on what stage and what kind of disease you are targeting. As I showed in my slides, that cells as a scaffold completely integrate, whereas when they inject it as a suspension, the integration may not be as efficient. It will be there, but it may not be as efficient. So it really is a matter of efficiency of integration and how fast you want the cells to start functioning, because as they're transplanted in a monolayer, the whole monolayer we have tested is already functioning, so they should be kind of, so to speak, hit the ground running. Whereas if they're injected as a suspension, they will need time to integrate into the monolayer before they can start functioning. So it really depends on the stage and kind of disease you are targeting. The approach might be different.
Great, thank you. Okay, so we just have one other question here about modeling of AMD. So is there mathematical modeling of AMD